Hi all. So today we're going to talk about protein synthesis. And this is pretty important. This is why DNA is so important. It kind of stores the information for how to build proteins. And those proteins are what give you your traits. So sometimes you'll hear this referred to as the central dogma or central idea of genetics, this kind of building blocks for the concepts of genetics. So if you can understand how this works, then you can figure out what's going on when it messes up. And the big idea here is that you go from DNA to messenger RNA to a polypeptide, which is kind of a fancy way of saying protein. And that polypeptide is going to result in a trait. So you can think like eye color, whether or not you can digest certain foods. And so let's look a little bit more closely at this whole kind of idea. So first you go from DNA to messenger RNA. And that's all going to happen inside of the nucleus. Now remember, DNA is going to be a double-stranded molecule. It's your master copy of all the instructions that you would need to build an organism, or all of the proteins in that organism. And you don't want to mess that up, and you don't need all of it all the time. So this first process of going from DNA to messenger RNA, you're going to copy a short section or a gene from the DNA into your working copy or messenger RNA, which is a single-stranded RNA molecule. And this process is called transcription. So transcription occurs in the nucleus. DNA is copied into messenger RNA. And now that messenger RNA meets up with a ribosome and some transfer RNA, and all three of these work together to go from messenger RNA to a chain of amino acids. So each of those would be like an amino acid. You put those together to form your polypeptide, which could become a protein. And this process is translation. So transcription is the first stage, translation is the second stage. Translation occurs out in the cytoplasm. Right, so out here in the cytoplasm. And it requires kind of all three of these RNA types to work together. A couple of things to make it easy to remember which one comes first. So transcription, I always remember transcription with the C. Comes before translation. With the L. C comes before L. Which is kind of silly, but when all the words just sound the same, sometimes you have to come up with little tricks to remember. Alright, so let's look a little bit more closely at each of those kind of stages. This is a big kind of overview. So, transcription is where you go from DNA to messenger RNA. It occurs in the nucleus. And what happens is a gene, or a section of DNA that codes for a protein, so let's define that, gets copied into messenger RNA. And that messenger RNA is kind of like the working molecule. It can go out and be read by a ribosome to create your protein. So we've got transcription, gene, and so you're basically making an mRNA copy of a gene. You're going to need an enzyme, so there's an enzyme called RNA polymerase. Makes a copy by base pairing to the DNA gene. So it's going to synthesize So, if we kind of look at how this works, the first stage is going to be that you unzip the DNA. So the DNA unzips when this RNA polymerase, right, your RNA polymerase, is going to bind to a promoter region. So a promoter region is just a section of DNA that says like, hey, come over here, I need you to make this gene. Once it does that, so once it kind of binds onto that promoter region, then it's going to work its way down, creating mRNA by base pairing to the DNA template. So we call it a template. So remember that RNA is slightly different than DNA, right? So in RNA, has a U instead of a T. So let's do a little bit of practice with this whole transcription translation thing. So let's say I have a DNA strand that has ATG, CGT, AGG, TAA. So the first thing I want you to do is copy that down. Then I want you to write in the complementary DNA strand that would go to this DNA strand here. Pause me and write it in. Then when you're done, come back and unpause and see if yours matches mine. All right. Glad you're back. So let's see. A goes to T, T goes to A, G to C, C to G, C, A, T, C, C, A, T, T. So this is just normal DNA to DNA base pairing rules. Well, now let's use this as our template, our gene, right, that we want to copy into the messenger RNA. All right, well now we need to write in the RNA or messenger RNA that would be created. So again, pause me, practice, see if you can do this for yourself. And when you're done, come back and we'll check it. Remember, RNA has U, not T. Okay. So now let's check it. So T still base pairs to A in RNA, but A is going to go to U. C goes to G, C, G, A to U, A, G, G, U, A, A. Sometimes this takes a little practice, so if you need to go back and review, that's fine. So now this messenger RNA strand is going to detach and go off to the cytoplasm for translation. So now let's look at translation. All right. So translation occurs in the cytoplasm. Here, we're going from messenger RNA to protein. If we want to be more specific, it's actually a polypeptide because some proteins consist of multiple chains of amino acids. But in general, we're saying messenger RNA to protein. And there's a few different players involved here. First, you have that messenger RNA, and it has the instructions. In addition to that, you're going to have your transfer RNA, and tRNA is going to carry the amino acids. And these are important because you need these to build your protein or polypeptide. So this is your amino acid. And then last but definitely not least are our ribosomes. And the ribosomes actually put the amino acids together. So let's go back and look at that messenger RNA that you just transcribed. You had A, U, G, C, G, U, A, G, G, U, A, A. Now, this messenger RNA is going to be read in 3 base words called codons. So each of these 3 base words is called a codon. So you may want to write that up here and remember that this has the codon. And each of those codons codes for one specific amino acid. Now the codon tells you what amino acid, but the transfer RNA is the one that has to bring it over. And it actually has its own kind of RNA code that base pairs with the messenger RNA. And we call that area is right over here, and it'll be 3 bases. And that's going to be called the anti-codon. It's not like against it, it just is the complementary base pair to it. So if AUG is my codon, my tRNA anticodon would be UAC. And so I would have, my little tRNA come over carrying a specific amino acid that was called for by that AUG. So pause me again, go in and see if you can fill in the other anticodons. All right. So I have G, C, A, U, C, C, A, U, U. To think about that sometimes. And each of these is carrying an amino acid. Well, how do I know what amino acid it's carrying? That's kind of important, right? Because this is the important part of the DNA, or the important part of the whole thing is this code tells you what amino acids to use to build the protein. Well, in order to do that, in order to figure that out, you need a codon chart. That's kind of like your own personal decoder ring. So pull up or take out your codon chart, and there's different types of codon charts. A couple things I want to point out. These are messenger RNA codon charts. So these only work with mRNA. They don't work with the tRNA, they don't work with DNA. They are coded for the messenger RNA codon. And I'll show you kind of how they work, and then you can do some practice. So I'm just going to zoom in here real quick. I'll try to. There we go. All right. So let's look at my first one. It's AUG, right? So AUG. I can look on this one, and on these kinds, you start in the middle and you work your way out. You can also use this kind over here. And there you 1, 2, 3. So let's start with this one. So I've got A in the center, U, G. So AUG, I get methionine or MET. So that first tRNA is going to bring over an amino acid called MET. And this is actually always going to be the way that a protein begins. This is what we call the start. All right, well, let's take a look at the second one. So here we have CGU. Again, let's take a look over here. CGU. So this one, my first base is C, so it has to be something in this whole area here. My second base is G, so it's going to be something in this one here. And then my third one was U, so it's going to be this guy right here. And I probably should have used a lighter color, but this says ARG, ARG. Really, it stands for arginine. So this is going to bring over ARG. All right, so pause me again, go in, see if you can figure out the other two. Remember, you're using the mRNA. All right, so I've got AGG, AGG, ooh, ARG again. So one of the interesting things you'll see here is that they have some redundancy in the code. So even though each of these codes for a specific amino acid, multiple codons can code for the same amino acid. We call it redundancy. All right, let's look at this next one, UAA. UAA, it says stop, and that's exactly what you do. So what happens is once you reach a stop codon, the ribosome is going to detach, and you're going to let go of this newly formed polypeptide. The ribosome, by the way, comes through and it makes the connections between all of these. So that was transcription and translation. We'll do plenty of practice on this, but hopefully that was a useful overview.